Welcome. We are currently at Myeloma 2018 in San Diego, and we've recently finished a session talking about new drug targets in myeloma. And I'm pleased to welcome my excellent panel, Drs. Bergsagel and Stewart from the Mayo Clinic and Dr. Orlowski from the MD Anderson. Maybe if I can start with you, Leif. We were in the discussion, we were talking about lots of new targets, but lots of familiar targets with potentially therapeutic options. Can you tell us a little bit about your work?
Well, that's a really good question. There's some old targets that we know about in myeloma that are probably still as important today as the day we discovered them, but we don't know how to target them, or they're called undruggable. And that would be something like cyclin D1 or MAF or MMSET genes that are dysregulated by translocations. And another gene in that class is MYC, which is important not just in multiple myeloma, but a lot of cancers. And we don't have a drug that can inhibit it. But a recent discovery is that we can inhibit what turns it on. And we do that by targeting the enhancers that regulate its expression. And there's a new class of drugs called bromodomain inhibitors that are able to do that. And there was discussion today about how we could maybe direct those drugs, which affect enhancers everywhere in the body, maybe to focus in on the enhancers that are really most important for multiple myeloma by combining them with other drugs we use in myeloma, like the IMiDs.
So you were particularly saying that the MYC and a BET domain inhibitor was going to be hopefully effective.
By using very low concentrations of each drug, you can get excellent therapeutic effects and overcome at least some of the resistance that develops to the IMiDs in patients.
Excellent, okay. And Keith, you were talking about some of the new targets that we could maybe go after.
Well, I was impressed with the number of new targets that were discussed during the session. Dr. Anderson talked about new ways of targeting the proteasome. We heard about targeting the BCL pathway, particularly targeting MCL1 as an exciting new target with multiple drug companies entering clinical trials. And we heard about some targets that are being identified in the lab. One we talked about ourselves was Pykfvye as a potential target. There are some drugs that target Pykfvye already in clinical trials around the country. So it was quite encouraging to see that we're not stuck with all the old targets that we had before, but there's some new things emerging. And I know Dr. Orlowski will talk in a minute about one of the most exciting new classes, which target, really you can target any protein you want using certain chemistries. So we heard about that. We also touched on immunotherapy. We'll hear a lot more about that tomorrow. But in our panel debate, there was the question of, or is there still room for small molecules targeting new targets in the immunotherapy era? And the conclusion I heard was yes, but probably we have to combine those therapies together.
Okay, there was quite a lot in there as well. Venetoclax kept coming up and BCL2. Do you want to expand a little bit on that?
Well, yeah, so venetoclax is very active in multiple myeloma. We see that already, that 40% of patients will respond at least, and if you concentrate the patients for those who have a translocation 11-14, you can see response rate up to 80% of patients. Unfortunately, not durable in all patients. There does seem to be resistance mechanisms emerging, and we heard a little bit about that today. And then we also heard about targeting other elements of that pathway with drugs which target MCL1 and even combining those together to try and overcome resistance. So we're all quite excited about building on venetoclax, which we think is heading for some kind of regulatory approval for use in myeloma with some of these newer agents.
And Keith mentioned there a little bit about not only the new targets, but some of the new, I guess, chemistry and drug methods for making drugs against these targets. Could you expand a little bit about that?
Sure. I think there's probably two new pathways or mechanisms that look interesting. First of all, there are antisense molecules, which are pieces of RNA, if you will, that can be used to target certain genes and proteins that are very specific, and some of these are now showing activity in myeloma models in the laboratory and are probably going to be coming out to the clinic. And those are very exciting because if you pick the sequence of the target correctly, you can inhibit just one protein from being produced. And then the degraders that Keith mentioned, are a way to do the opposite of what we're used to doing, which is proteasome inhibitors, which block protein digestion. But there is now technology that allows you to actually activate digestion of proteins very specifically, so that if you have a protein that is responsible for a bad behavior in myeloma, and MYC could be one of them, you can use the technology to degrade MYC and maybe a small number of other proteins, but it's much more targeted and much more specific. The problem will be to make sure that we target proteins that are important to myeloma and not to normal cells, because then there will be toxicity.
I guess one of the other things we'll need to think about as well with those new drugs is maybe it's not going to be automatic that we can combine them with some of the drugs we already use.
Correct, because the digestion of the proteins occurs through the proteasome. We're not going to probably be able to combine them with proteasome inhibitors, for example. But hopefully the benefit will outweigh the downsides.
Yeah. And then Leif, in the kind of panel discussion, I think Dr. Anderson brought up that at the moment many of our targets are just going after the myeloma cell, and that maybe we needed to think a little bit about the microenvironment.
Yes, Dr. Anderson raised the point that the drugs we're using aren't just targeting the tumor cell, but they're also having effects on the microenvironment. And that becomes particularly important when we think about combining them with immune modulators. If our drugs either inhibit or augment those activities. And he described some examples of what he thought were promising ways that we could move forward with those, with both drugs that are called checkpoint inhibitors, which could be explored further using new kinds, and also drugs which can augment their activity.
And then, Keith, maybe just finally to close, there was a few controversial comments there about the fact that actually we don't need to do targeted therapy anymore, and that immune therapy was going to solve everything. Thoughts?
Well, we could only hope, but I think it's early days for immune therapy. Clearly people do relapse after that treatment, although hopefully some are cured. For those who do relapse, we're still going to need new targets, new therapies. There's also a cost issue. These are very expensive therapies, immune-based treatments, particularly using T cells. And there may be areas in which a lower cost but as effective small molecules could either replace or synergize with the immune-based therapy. So I think until we cure the disease, we still need to work on all available options.
No, I think I agree. And I think it was a really good session. It was nice to see we weren't just talking about things that are going to happen in the next 5 years. Many of these things are going to be coming to the clinic in phase 1 trials in the next year or so. So thank you very much for sharing.